Thank you for the thank you for arranging this. My name is Arvind Mer, and I'm assistant professor at University of Ottawa. I was intent to attend in person. However, I got COVID because I was traveling to another conference and I got COVID and I could not attend in person. But anyways, title of my talk is Enhancing Machine Learning Based Drug
So
Response
can you increase the volume a little bit more?
Okay.
Is that possible?
Try. Oh, it seems maximum from.
I'll try to make some adjustments. Give me one moment.
Okay.
That is better.
Is it better?
Yes. Okay.
Okay.
Yeah.
So should I start from the beginning or it's okay?
Yes, from the beginning
would be great. Okay, great.
We do not see your slides.
Oh, let me share again.
Sorry for the technical difficulties, everyone.
No.
You did have to switch this one.
Share. Do you see it now?
No, we do not.
Oh.
So if you go into the bottom left, you should see a thing that says share. Make sure you go to the screen itself.
See the slide on the bottom. Maybe you're in the full screen mode. That's the reason.
Oh, there we go. Okay, sorry about that.
Okay.
It was my error. My error. You're set up now.
Okay. Great. Thank you. Okay. Hello, everyone. My name is Arvind, and I'm assistant professor at University of Ottawa. I was planning to attend this exciting meeting in person. Unfortunately, I got COVID a few days ago as I was traveling to another conference, and that ruined my plan. I would like to thank organizers for giving me opportunity to present my work online. So my title of my talk is Enhancing Machine Learning Based Drug Response Prediction Models via Text Mining Driven Feature Selection Approaches. So in general, what we do in my lab is that we are a computational biology bioinformatics lab, and we look into the omics data, especially in context of cancer, and we try to analyze this omics data to build machine learning models that hopefully can find responders from non-responders in setting of the cancer. So this is where our main analysis is. In practice, what we do is that we have a lot of genomic data as well as drug response data that we analyze. We build tools and databases to manage this data, as well as build machine learning models on the top of these. So as many of you know, when you do machine learning, a big challenge is the feature selection, especially in context of biology. So we have a large number of features in biology. For example, from RNA-seq, you get almost 22,000 genes, and then you want some feature selection approach that can reduce these features or select the features that are most relevant to your problem, and then build the model on those based on those features only. So there are many different feature selection approaches exist there in machine learning field, wrapper-based, embedded methods, or filtering methods. They have their own advantages and disadvantages. Some are suitable for certain cases and not suitable for certain other cases, and so forth. So people are also using advanced deep learning approaches for feature selection, and but the big problem is that they are many times black box kind of models, and it's really difficult to understand which features are being selected and why they are being selected. Similarly, there are hybrid approaches that mix different feature selection approaches. So especially in context of the modeling the pharmacogenomic, cancer pharmacogenomic data, we see a lot of papers that are showing that different feature selection approaches, feature selection approach X is performing better than Y and so forth. And I always wondered that do we need these complicated approaches? Are there simple approaches can perform similar or not? So the simple question we asked, that can a text mining based feature selection approach outperform other approaches? So what I mean by text mining based feature selection here is simply, for example, this is one of the abstract related to a drug called erlotinib, the anti-cancer drug. And as you see, erlotinib is mentioned with the gene EGFR. So it's very frequently mentioned with this gene in EGFR in the literature. So there must exist a link between them in the real world. So by text mining genes, I mean text mining features, I mean those genes that are mentioned in the literature near to or near to a drug or any term. So to find these text mining based features for these drugs, we used an existing algorithm, Genie. This was published by Miguel Andrade back in 2011. What this approach does is that if you give it any term, like for example, in our case, name of a drug, it just goes to the PubMed and looks into all the abstracts and finds that term and looks which genes are mentioned near to those particular terms. The term of interest, and by this way it can make a list of all the genes that are relevant to that particular term. In our case, we are using drug as a term, so given a name of a drug, it gives you all the list of a gene that are associated with that drug. So this is our analysis pipeline in context of comparing this text mining approach versus other approaches. So our training data set is CCLE, Cancer Cell Line Encyclopedia, which contains large set of cell lines as gene expression from cell lines, as well as drug response on those cell lines. That is our output. We are using several different feature selection methods for comparison. That includes L1000, top 500 variable genes, different wrapper methods, and deep learning methods also. In terms of machine learning algorithm, we used 3 different machine learning algorithms: random forest, elastic net, and deep learning. So using these different, using these feature selection method and CCLE data, on CCLA data, we select features, we build machine learning models using these 3 different methods, and then we test those models using an independent data set called GDSC that also contains drugs and gene expression from the cell lines. So we use it as a validation and then we compare it. So here are some of the results when we look into the drug. So in the first figure on the y-axis, you see different name of different drugs, and the x-axis, you see a number of genes that we found using the text mining tool. So there's a large variance between these different drugs. Some drugs have a lot of genes have been associated with them, while some of them has only very few genes associated with them. And when we look how these genes are distributed, as expected, some of the genes such as EGFR, because they are kinase inhibitors, etc., KRAS, etc., are associated with lots of drugs, and they are very commonly present in the literature. When we look at the univariate analysis, where we simply compute the correlation between expression of a gene and the drug response and divide them by text mining versus all other genes, we found that in case of text mining, there is a significantly high correlation is there between the gene expression of a text mining based gene and the drug response compared to the other genes. In terms of machine learning, as I mentioned earlier, we used 3 different machine learning approaches. So these are results for the random forest-based machine learning algorithm. So the first plot shows the training results. As you can see, is the text mining based genes, and all the other different models, different feature selection methods are here. We use the t-test for the comparison here, and the text mining is outperforming all the methods in this scenario. The bottom panel shows the random forest validation. The text mining outperforms few of them very well. However, in case so, we do not find any statistical significant difference in case of MRMR and recursive feature elimination method. Using the elastic net model, we found even better results where text mining based models, text mining based feature selection models outperform all of the methods in the test data set, in the independent test data set. Moving forward, we applied the deep learning also, and the deep learning in learning this method also.
Arvind, sorry to interrupt, but we're running behind. Would you be able to wrap up, please?
Yes, just 2 minutes. Just in the case of deep learning based methods, we found that it outperforms text mining approach, outperforms all other methods. We also validated this using in vivo data, where we found that our text mining based approach model can build machine learning models that are very transferable to other data types also. So to summarize, we have shown that text mining genes outperform other feature selection approach. There are certain limitations. We are only looking into the limited scientific literature, and in future we want to optimize these to select the better features. Okay, and this work has been published in Bioinformatics Advance. And if you have any questions, please feel free to ask me. Thank you.
Because we are running behind, we have one quick, maybe burning question. Otherwise, we'll move on to the next talk. Thank you again, Arvind.
Thank you.
Go ahead.
Hi. So it's a really nice work. I was just wondering, what are your thoughts on potential data leakage, knowing that these public databases that you use, like let's say GDSC, also have publications that might have been, or citing publications that might have been in the corpus that you used.
Yeah, that's a really good and interesting question. We haven't looked into this specific problem, like how these two data sets are affecting each other. But the thing is that most of the gene lists that we are getting, or genes we are getting, are from the classic biology experiment papers, not from the big papers. So it rarely happens that GDSC or CCLE and all these papers mention specific genes, because their focus is more broadly. So I cannot say that there is no such data leakage, but my belief is that it's minimum in this case, at least.
Thank you so much.